Hello students, welcome back to Target Prelims 2026 Revision with MCQ series, and this is a new episode of Science and Tech. So let's begin with the first question for today. With reference to the National Automated Fingerprint Identification System, consider the following statements. 1. It is a pan-India searchable database of crime and criminal-related fingerprints. 2. It was developed by the National Crime Records Bureau. 3. It assigns a unique 10-digit national fingerprint number to each criminal which remains valid for the lifetime of the offender. How many of the statements given above are correct? A. Only 1, B. Only 2, C. All the 3, or D. None. So please take a few seconds, attempt this question, and then we'll discuss the explanation. Okay, so we are talking about NAFIS, National Automated Fingerprint Identification System. So this is actually a digital system. It's a digital system which stores the digital fingerprint of all the criminals. So it's done once only and it remains valid for the lifetime of that criminal, lifetime of the offender. It's particularly helpful in taking care of repeat offenders because we have it in the database. Their fingerprint is already present in our database, so it will be easier to identify the criminal, any sort of a repeat offender. And if there is a link that has to be drawn between multiple cases, at times there are such cases where they are linked. So in those cases also this database is especially helpful because we have the digital fingerprint of that criminal, right? So it's a pan-India searchable database of crime and criminal-related fingerprints. Yes, this statement is correct. It was developed by the National Crime Records Bureau. Yes, it was developed by the National Crimes Record Bureau, and it stores the fingerprint number, which is actually a unique ID. It's a 10-digit national fingerprint number to each criminal. It is assigned to each criminal and it remains valid for the lifetime of the offender. Okay, so a criminal's fingerprint is taken, digital fingerprint is taken, and then a 10-digit number, a unique ID is assigned to that criminal and it remains valid for the lifetime of that criminal. That's why it is going to help us in deal with repeat offenders, right? So the correct answer over here would be all the 3 statements are correct. Option number C. So you will have the PDF. You'll have this PDF on our Telegram channel. Please go ahead, download it from there, and there you can look at the explanation. Okay, moving on to the second question. Consider the following statements about the Global Antibiotic Research and Development Partnership. 1. It is a not-for-profit organization that develops new antibiotics to combat drug-resistant bacterial infections. 2. It was created by the World Health Organization in collaboration with the Drugs for Neglected Diseases Initiative. Which of the statements given above is or are correct? A. 1 only, B. 2 only, C. both 1 and 2, or D. neither 1 nor 2. Please attempt this question. Okay, so we are talking about the Global Antibiotic Research and Development Partnership. It is a partnership. So let's break it down first. This is a global level partnership. It is a not-for-profit organization which has this partnership. It was done in collaboration basically. And here we are talking about an independent body. So this partnership over here, this body, it is an independent body. Okay, it's an independent body and it helps in the development of new antibiotics, particularly in those cases where we have drug-resistant bacteria. So many a times we see that bacteria develops a resistance for all these different types of drugs, right? So we have multi-drug resistant bacteria, extremely drug resistant bacteria. So we have these different strains of drug resistant bacteria, and dealing with them or treating them is especially difficult. So for that we have this body, this partnership that helps in the development of new antibiotics, particularly for dealing with drug resistant bacteria. And the goal over here is to make such medicines, make such drugs actually affordable and accessible to all, right? So it's a not-for-profit organization that develops new antibiotics to combat drug-resistant bacterial infections, right? Now, this was developed as a partnership. It was created by the World Health Organization in collaboration with the Drugs for Neglected Diseases Initiative. Yes, this statement is also correct. So, both the statements over here are correct. Correct answer is option number C, both 1 and 2. Okay, moving on to question number 3. With reference to the National Supercomputing Mission, consider the following statements. 1. It is a flagship initiative launched to promote self-reliance in supercomputing technologies. 2. The mission is jointly steered by the Department of Science and Technology and the Ministry of Electronics and IT. 3. The Trinetra network developed under the NSM supports data transfer speeds up to 2 terabits per second. Which of the statements given above are correct? A, 1 and 2 only, B, 2 and 3 only, C, 1 and 3 only, or D, 1, 2 and 3? Please attempt this question. Okay. So the National Supercomputing Mission wants to build these supercomputers indigenously. So the goal over here is to build high-speed computers that can, that can be very effective in doing some very high-speed calculations, and we want to do that indigenously. We want to, reduce our reliance or reduce our dependency on other countries where we are dependent on other countries' supercomputers. Instead, we want to build our own supercomputers. So for that, we have the National Supercomputing Mission, and this mission is jointly steered by the Department of Science and Technology and the Ministry of Electronics and IT. So this is led by this department as well as this ministry. But when we talk about the implementation, it is implemented by CDAC Pune and IISc Bangalore. CDAC Pune and IISc Bangalore. Let's look at the first statement. It is a flagship initiative launched to promote self-reliance in supercomputing technologies. Correct. This statement is true. Then the mission is jointly steered by the Department of Science and Technology and the Ministry of Electronics and IT. This statement is also correct. And now we are talking about the Trinetra network. So Trinetra network basically is a high-speed network. In order for us to transfer data in this National Supercomputing Mission also, we are building a very high-speed data network where data can be transferred very effectively in a very speedy manner. So that is the Trinetra network. So this network developed under NSM supports data transfer speeds up to 2 terabits per second. So this is actually incorrect. It is 200 gigabytes per second. So if you look at the explanation over here, we will see that the Trinetra high-speed network developed under NSM boosts data transfer rates up to 200 Gbps, gigabytes per second, and it is being rolled out in phases. So we have these phases, these different phases. That's why the third statement is not correct. So the correct answer would be statement 1, statement 2 are correct. Option A, 1 and 2 only. Okay. Moving on to question number 4. With reference to the Vikram-I launch vehicle, consider the following statements. 1. It has been developed by Skyroot Aerospace, a private Indian space startup. 2. It is a 4-staged orbital launch vehicle designed to provide quick and cost-effective launches for small satellites. 3. The rocket incorporates 3D printed engines to reduce weight and manufacturing time. Which of the statements given above are correct? A, 1 and 2 only, B, 2 and 3 only, C, 1 and 3 only, or D, 1, 2 and 3. Please attempt this question. Okay, so Vikram-I is a launch vehicle. Vikram-I is a launch vehicle whose goal is to place small satellites to the orbit. Okay, placing small satellites into the orbit. Now this has been developed by Skyroot Aerospace, a private Indian space startup. Yes, this statement is correct. It is actually being developed, this launch vehicle. It was one of the milestones that we achieved since the commercialization of the space sector in 2020, where we opened up the space sector for private players also, for private vendors also. So this was a private launch vehicle that has been developed by Skyroot Aerospace. Then it is a four-staged orbital launch vehicle designed to provide quick and cost-effective launches for small satellites. So this statement is also correct. It is indeed a four-staged vehicle, and if we talk about the stages over here, so the first three stages, 1, 2 and 3, the first three stages have solid fuel, okay, to give that powerful lift off, that thrust. So we have in the first three stages we have the solid fuel, and then the game changer is the fourth stage, where we have a liquid fuel. Liquid fuel makes controlling the device very easy, controlling the components very easy, maneuvering becomes easy. So in the fourth stage we have liquid fuel, but in the first three stages we have a solid fuel. Okay, so this statement is also correct. Then the rocket incorporates 3D printed engines to reduce weight and manufacturing time. Yes, this is also correct. It is a very advanced rocket. It actually has made use of 3D printing technology also. 3D printed engines are being used, and with that the weight has also reduced. It has become more lightweight, and the manufacturing time has also reduced. So it has become more efficient in nature. Okay, so the correct answer would be, all the three statements are correct. Option number D, 1, 2 and 3. Moving on to question number 5, with reference to Bharani MK1, consider the following statements. 1. It is designed for surveillance of low and medium altitude aerial targets. 2. It is highly mobile and can be transported by vehicles, animals, manpower, or as a heli-slung cargo in difficult terrain. Which of the statements given above is or are correct? A. 1 only, B. 2 only, C. both 1 and 2, or D. neither 1 nor 2. Please attempt this question. Done? So this is about Bharani MK1. Bharani MK1 is actually a lightweight radar system. Okay, so this is a radar system. This is lightweight, and it is used to detect all sorts of aerial threats. Okay, all sorts of low flying aerial threats, like you could have drones. The use of drones is becoming very prominent these days in the modern warfare. So we have drones that can be detected, we have helicopters that can be detected, or any other low flying aerial targets that can be detected by the help of this lightweight radar system. Okay, so that is Bharani. Again, it is lightweight, it is portable, it is very easy to transport it from one place to another, to take it from one place to another. So you can, it's specially helpful in difficult terrain, right? In rocky terrain, in difficult terrain, in different altitudes where heavy systems cannot be transported. This is very easy to transport. So the first statement says it is designed for surveillance of low and medium altitude aerial targets. Yes, this statement is correct. It is for low and medium aerial targets like your drones, like helicopters, or any other low flying aircrafts. It is highly mobile, correct, and it can be transported by vehicles, animals, manpower, or as a heli-slung cargo, even through the helicopters, in difficult terrain. Yes, this statement is also correct. That actually is the USP of this radar system, that it is lightweight, it is portable, and it can be used in difficult and rocky terrain also. Okay, so both the statements are correct. Option number C, both 1 and 2. Question number 6. Consider the following statements regarding the quantum diamond microscope. 1. It is a quantum imaging system that can optically map dynamic three-dimensional magnetic fields at the nanoscale. 2. It has been developed at IIT Bombay under the National Quantum Mission. 3. It can be used for biological and semiconductor studies. How many of the statements given above are correct? A. Only 1, B. Only 2, C. All the 3, or D. None. Please attempt this question. So quantum diamond microscope. Quantum diamond microscope is actually a very advanced microscope that can detect magnetic fields, tiny magnetic fields also, by making use of the NV defect pair. So this microscope, this quantum diamond microscope, this actually uses quantum physics. Okay, it uses the principles of quantum physics, and it can detect very tiny, very small magnetic fields in particles as well. So what happens is that generally when all the carbon atoms are present, they are bound together, then in one of the cases, in cases of this kind of a quantum diamond microscope, we see that one of the carbon atoms is missing. One of the carbon atom is missing and a nitrogen atom is found next to that carbon atom. So what we get is a nitrogen vacancy defect pair. Vacancy which has been created because of the absence of the carbon atom. So because we have this defect pair is actually present, and this defect pair is extra sensitive, super sensitive to all sorts of magnetic fields. That's why it is able to detect magnetic fields at such a nano level also. Okay, so it's a quantum imaging system that can optically map dynamic three-dimensional magnetic fields at the nano scale. Yes, this statement is correct. And an interesting point over here is that it can do so even at room temperature. It's making use of quantum physics, and when we talk about devices or things making use of quantum physics, they most of the times require very cold temperatures. They're very fragile systems, they require very cold temperatures and cooling systems and everything. And because this can work even at room temperatures, it's very cost effective. You don't have to, it's easy to maintain. You don't really have to maintain a cooling system at all times. It's not that fragile, it's more stable. So these are certain things that make this microscope even more important. Okay, so it has been developed at IIT Bombay under the National Quantum Mission. Yes, this statement is correct. And then the third one says that it can be used for biological and semiconductor studies. True. It has various applications because it can detect magnetic fields at nanoscale. These magnetic fields or this nanoscale type of study can be done everywhere. It can be done on inanimate things also. It can be done on biological things as well. So biological field also, this has an application. And when we talk about semiconductor studies, there also it has an application. So here all the three statements are correct. Option number C, all the three. So the quantum diamond microscope is an advanced quantum imaging system that uses nitrogen vacancy centers in diamond to optically map dynamic three-dimensional magnetic fields at the nanoscale, right? Diamond, you have all these carbon atoms bound together. One of the carbon atoms, when that goes missing, the nitrogen atom is present next to it, then it creates a nitrogen vacancy center. It's a defect pair. This is a defect pair, and this defect pair is super sensitive to all sorts of magnetic fields that are found at nanoscale. Okay. Question number 7. With reference to nanoenzymes, consider the following statements. 1. Nanozymes are nanomaterials that can mimic the catalytic activity of natural enzymes. 2. They can help regulate reactive oxygen species levels and reduce the risk of abnormal blood clotting. Which of the statements given above is or are correct? A. 1 only, B. 2 only, C. both 1 and 2, or D. Neither 1 nor 2. Please attempt this question. Okay, so we're talking about nanozymes or nanoenzymes. So firstly, let's understand what are enzymes. Enzymes are what? They are catalysts. They speed up the reactions. They speed up reactions and they are natural. So enzymes are natural catalysts, right? Now when we talk about nanozymes, nanozymes are not natural, they are in fact artificially engineered, they are artificial, they are made in laboratories and they are the artificial version of these enzymes. Okay, so enzymes are found naturally in our body, they are catalytic in nature, they speed up the process, they speed up the rate of any process that is going on in our body. Then when we talk about nanozymes, nanozymes are artificially made, they are artificially lab manufactured, and these can mimic what the enzymes do in our body. Okay, so nanozymes are nanomaterials that can mimic the catalytic activity of natural enzymes. Yes, this statement is correct, and they can help in regulating the reactive oxygen species level and reduce the risk of abnormal blood clotting. Now let's understand this. What is reactive oxygen species level, or the ROS levels? When we talk about oxygen molecules, if you have ample amount of oxygen molecules, like normal level of oxygen molecules in the body, then it is good for the body because that is required for the proper functioning of the body. But when we have a lot of oxygen molecules, when we have a lot of oxygen in the body, then it starts getting reactive in nature. Okay, it starts getting reactive in nature, it starts making other cells of the body overactive and it starts affecting their functioning. So let's understand this in the form of blood platelets. So when you have ROS in limited quantity, reactive oxygen species, when you have that in limited quantity, then that works fine, that is okay, it's good for the body. But when we talk about it being present in abnormal levels, they start making the cells of the body overactive. When we talk about platelets, so the role of platelets here is to clot blood, right? Whenever there is an injury, blood clotting needs to be done so that loss of blood does not happen from our body. In that case, platelets play an important role. Now when we talk about high levels of ROS, high levels of reactive oxygen species, that means that the body is under an oxidative stress, and these platelets are also becoming overactive. They are becoming overactive and they are clotting even when it is not required. So the job of platelets is to clot the blood wherever there is an injury, wherever there is a site, something has happened, a cut is there, a wound is there, then it has to go and clot blood. But if it starts getting overactive, hyperactive, in that case what's going to happen, it will just start clotting blood everywhere. That is going to be harmful for the body, right? And it can also lead to death. So that's a dangerous situation, and in those cases we need the help of such kind of enzymes. Nanozymes, artificially created, then we can inject them exactly at the site where we need them to be. So we can direct them that this is the site, this is the particular area where they have to perform their catalytic activity. So they help regulate the ROS levels and reduce the risk of abnormal blood clotting also. Okay, so this statement is correct. Both the statements over here are correct. Just to give you one more example over here. So by controlling excess ROS, these nanozymes prevent overactivation of platelets, thereby reducing abnormal blood clot formation, such as in pulmonary thromboembolism. So this happens in the lungs, where the abnormal blood clotting starts taking place, and that can also, in chronic situations, it can also lead to death. So among various materials, if we see some of the materials or some of the nanozymes are spherical vanadium pentoxide. Spherical vanadium pentoxide nanozymes were found to be the most effective. They mimic the activity of glutathione peroxidase, a natural antioxidant enzyme that reduces the oxidative stress. Okay, so oxidative stress is happening when too much of oxygen is present in the body, too much of like the ROS levels are very high. In that case, these cells, they start becoming overactive, they perform more, and at times it is going to lead to difficult situations for the body, right? So now let's move on to question number 8. With reference to extrachromosomal DNA, consider the following statements. 1. ecDNA are small circular DNA pieces present in the nucleus. 2. ecDNA can form due to chromosome breakage or errors during DNA replication. 3. ecDNA often contains cancer-causing genes and supports tumor growth and drug resistance. How many of the statements given above are correct? A. Only 1. B. Only 2. C. All the 3. Or D. None. Please attempt this question. Okay, so extrachromosomal DNA, as the name suggests, is found outside the chromosome, within the nucleus but outside the chromosome. Okay, outside the chromosome. When we talk about DNA, so DNA is tightly packed within the chromosome inside a nucleus, and when at times when this chromosome or this DNA is actually copying itself, it is making its copies, at that time some of the chromosomes can break. Okay, when some of the chromosomes break, they can actually move out. Some of the DNA can actually move out of that chromosome, and it starts floating, kind of floating inside the nucleus in a small circular shape. Right, so that is your extrachromosomal DNA because it is found within the nucleus but outside the chromosome. So ecDNA are small circular DNA pieces present in the nucleus. Yes, this statement is true. Now I told you that they are being formed by accident. It is not intended, but by accident, while the DNA is copying itself, some of the genes can break down. Some of the genes can actually break apart, and then they create this extrachromosomal DNA. Okay. So second statement says ecDNA can form due to chromosome breakage or errors during DNA replication. Yes, this statement is also true. Now problems actually arise when we talk about cancerous genes or cancer carrying genes, right? So I told you that this is being done by accident. When the DNA replication process is taking place, some of the chromosome is breaking, and if the genes that are breaking, if those were cancer genes or they were cancer carrying genes, then what are we going to get? We are going to get extra copies of those cancerous genes outside the chromosome also, right? Because what's happening over here, when we talk about the breakage of chromosomes, some of the DNA, while its replication, it is creating extra copies, the copies that were not intended, copies that were not supposed to be created. In normal circumstances, it is still okay. But if we talk about cancerous genes or cancer carrying genes, in that case, if we have them present outside the chromosome also, then what will happen? Treatment is going to get difficult, because now it will be difficult to identify the target. If we know that the DNA, all that cancer causing DNA also is present within the chromosome only, then targeting that becomes easier. But now it is going to be present outside also, in other areas also, and then it is going to replicate even faster, it is going to grow faster, and the tumor is going to spread because now it is present at different places. So ecDNA often contains cancer causing genes and supports tumor growth and drug resistance. Yes, this statement is correct. So all the three statements are correct. Option C, all the three. The extrachromosomal DNA consists of circular DNA fragments that exist outside the standard chromosomal structure within the cell nucleus, right? Advances in detection techniques, including microscopy-based and sequencing-based methods, have substantially improved our understanding how ecDNA contributes to cancer growth and evolution. They are formed due to chromosomal damage, the breakage that happens, catastrophic events like chromopsis or replication errors. In cancer cells, ecDNA frequently carries multiple oncogenes. Oncogenes are cancer-causing or cancer-carrying genes, promoting rapid tumor growth and drug resistance. So unlike chromosomal genes, ecDNA does not follow Mendel's law of independent assortment, as it is inherited in clusters during cell division. So it does not behave like normal chromosomal genes. It is actually caused because of an accident. Its behavior is also different, and then the treatment also gets different because the behavior is different. It is now present in other parts also. It is replicating faster, and it's creating, it's actually spreading the tumor faster. Okay. Question 9. Ricin, recently in news for security concerns, is best described as 1. A nerve agent synthesized from petrochemicals. 2. A toxic protein derived from cancer beans that inhibits protein synthesis. C. A radioactive isotope used in radiological weapons. D. A synthetic chemical used exclusively in industrial pesticides. Please attempt this question. Okay, so ricin was in news. Ricin was recently in news because it is a toxin. It is very poisonous. It is actually a toxin that is generated from castor beans. It is generated from castor beans, and the problem over here is that it actually it actually hampers protein synthesis. It does not allow cells to make protein. So once we are extracting oil from the cancer seeds, at times this toxic protein is also derived. This is ricin, and this inhibits protein synthesis in cells. So it does not allow cells to actually manufacture their protein or synthesize their protein, and without proteins the cells will die faster. They will die quickly. So this is a challenge over here, and a bigger challenge is that we don't have a specific antidote for this toxic protein. We don't have a specific antidote. Then what do we have? Only supportive care can be given. Once we see that this is a condition that a person has developed, we can only give that person quick medical attention, supportive care. But we don't have a specific antidote that is going to target specifically ricin and it is going to destroy it. We don't have that. That's why it's a very dangerous protein, a very dangerous toxic that is released once we are trying to extract oil from castor seeds. Okay. Moving on to the last question for today. With reference to the global virus network, consider the following statements. 1. It is a global coalition of virologists that works on understanding viral diseases and developing vaccines and therapies. 2. It was founded in 2011 by Robert Gallo in collaboration with international scientists. 3. It functions as a United Nations affiliated body responsible for global disease surveillance. Which of the statements given above are correct? A. 1 and 2 only, B. 2 and 3 only, C. 1 and 3 only, or D. 1, 2 and 3. Please attempt this question. Okay, so the global virus network is actually a very independent, very important body. It's an independent body, it's an important body, it's a network. It is a network of virus experts. Okay, it's a network of virus experts. Now, who are these virus experts? Virus experts would be your scientists, they would be your doctors. So these scientists, these doctors, they come together, they study the various viruses, the various diseases that are being caused by the viruses, new age viruses, everything, and then they help in the creation of, in the formation of medicines and vaccines. Medicines and vaccines to deal with the new types of viral diseases that are coming in. So these people, this is actually a group of experts who brainstorm, who do research, and then they help in the development of these medicines and vaccines. Okay. So this is a global coalition of virologists that works on understanding viral diseases and developing vaccines and therapies. Yes, this statement is correct. Now this was founded in 2011 by Robert Gallo in collaboration with international scientists. So this was the person, he was the person who pioneered the entire initiative in 2011. So this statement is also correct. and it functions as a UN affiliated body responsible for global disease surveillance. This statement is not correct because it is an independent body. This is an independent body. It is not a UN affiliated body. It is not a UN affiliated body. It is an independent body. It is a network of virologists, a network of virus experts, scientists and your doctors coming together, understanding the different viruses, the diseases caused by those viruses, and helping in the formulation of new medicines and vaccines. Okay, so 1 is correct, 2 is correct, 3 is incorrect. So the correct answer would be option A, 1 and 2 only. That was all for today's video. Now we will look at a practice question for prelims. With reference to SIM binding, consider the following statements. 1. SIM binding links a communication or messaging app to the SIM cards used at the time of user registration. 2. It has been mandated by the Department of Telecommunications under the Telecommunications Cybersecurity Amendment Rules 2025. 3. Under SIM binding, a user can freely access the app even if the registered SIM is removed from the device. Which of the statements given above are correct? A. 1 and 2 only, B. 2 and 3 only, C. 1 and 3 only, or D. 1, 2 and 3. So please attempt this question, provide your answers in the comment section, and we will meet in a new video. Thank you for watching.
For more informative content, like, share and subscribe, and do not forget to press the bell icon to get the notifications.